Thank you, Ton. It's actually wonderful to have heard the first half of this day because we saw so many things that were considered crazy ideas 10 years ago and not just came to fruition, but became mainstream. JBS Holden, the great biostatistician, said there are 4 steps for the acceptance of new scientific ideas. Step number 1, this is complete nonsense. Step number 2, this is an interesting but perverse point of view. Step number 3, this is true but unimportant. And step number 4, I've always said so. So I'm going to produce or present some ideas that are somewhere in the first or second step. A caveat, not everything that is considered uninteresting and unimportant becomes a well-accepted scientific view. Let's see if what we do today mature to that point. So that's my clicker. Okay, so last year, Nature celebrated the fact that the 100th therapeutic antibody was approved, and it reviewed what we already have, and people touched upon this point several times during the day. The most interesting fact here to me, as someone who studies proteins, is that virtually all approved antibodies are either antagonists or markers. They're not a single antibody that is an agonist in the classical sense of biological agonism. But, as you all know, antibodies are proteins, and proteins are nanorobots. They can be programmed. That's what evolution did. It programmed proteins to execute dynamic programs over time. But the pharma world uses antibodies as if they were small molecules that happen to be bigger, right? They're used to antagonize, maybe to block, maybe to mark. And the question is, can we unlock the great potential of antibodies as dynamic nanomachines and put it to work in the clinic? And what we are trying to do is to use artificial intelligence to do that. And artificial intelligence is a buzzword. Someone told me that the difference between artificial intelligence and machine learning is that artificial intelligence is something you write in PowerPoint, and machine learning is something you write in Python. So let's try and break it down to something more simple than that. Let's say a word about what are the capabilities we want to give to the antibodies. So let's start with simple things. We know that proteins and antibodies can be very specific in what they bind. They can bind a very specific site. They can bind a very specific conformation. We saw some examples for this today. Can we design that? Can we come up with precision binding? Another thing that we want to have is conditional function, and this is something that proteins do all the time. Can we have an antibody, for example, that in one tissue or under one set of circumstances will do one thing, and in another tissue or under another set of circumstances will do something else? Say, let's think about inflammation. So sometimes inflammation is caused by one cytokine and sometimes it's caused by another. Can we have an antibody that measures in each tissue and each patient and each point in time which is the dominant cytokine and antagonize just that? It's done in biology. Can we program antibodies to do this? So let's focus on 2 simple capabilities that every first-year student in biochemistry knows proteins have. And these are the precision binding and the dynamic multifunctionality. What is machine learning? Machine learning and artificial intelligence are fancy statistics. That's what they are. And hence, the key for success in machine learning is always the data. A typical stream for a machine learning project is to define what is it that we want to do. So it could be to read text, to identify cats in a picture. It can also be like AlphaFold to predict the 3D structure of proteins. Or it can be our task, make better therapeutics. Always, invariably, the first step is to find the right data, curate it. A lot of data, so it could be texts or pictures or 3D structures of antibodies in the PDBs. Then you train a model, or Ton says algorithms, and eventually you validate. Wonderful, easy. Where is the problem? The problem is that if our task is to design a better therapeutic antibodies, it's not very clear where we can get millions and millions of data points that are required in order to train a machine learning model. We can look at the protein world, but these are proteins in general. We can look at antibodies that were measured in the clinic to be excellent. There may be hundreds, maybe thousands, not enough data points to train a model. So that's the challenge for virtually anyone in the field. We know what we want to achieve. We have the challenge. We know how to validate it in the clinic. What we don't know is where to get the data. In Biologic Design, the company we founded a few years ago, approached this challenge with the following idea. Let's make our own data. So the platform that we set up begins with the challenge. So we don't leave it as an abstract notion of design better antibodies. We say the challenge is the TPP. What is it that we want the antibody to do inside the patient? It can be the biological effect, it can be the binding properties. Obviously, an important part of it, as we heard earlier today, is the developability profile. All these are what we want to predict. And then we need to get the data. And this is where our platform has a first step. This is model number one. Model number one tells us what data we need in order to be able to predict. So the first step is to analyze the data that we already have, everything we know about these challenges, and tell us what kind of measurements are missing in order to be able to achieve what we want. So if we want, for example, that antibody we mentioned before that can bind that cytokine in that tissue and another cytokine in another tissue, the model can tell us, well, we need more information about what allows a certain antibody to bind target X, and more information about what allows an antibody to bind target B, and then more information about what makes those developable. So this step is actually dictating a set of very focused libraries that we make, we go to the lab, and we do measurements for binding, for expression in these large-scale libraries with specific mutations that the model created. And typically at this stage, we generate about 10 to the 9th data points. This is enough data to train a deep machine learning model. Based on the data we collected at the second step, we can then train a machine learning model that predicts which sequence will do what we want. And then we can validate and continue to the developability. This model has been validated several times. The first computationally designed antibody, to our knowledge, is an antibody we designed and is now in phase 1/2 in cancer patients. I will tell you about it in a minute. We collaborated with several companies about challenges they could not solve with the discovery tools that they have. The capabilities that this platform allows us to do involve, for example, agonism by design, being able to agonize a protein by stabilizing the active conformation. Conditional functions, do if then, we'll show you some example for that. turning antibodies into sensors actuators, an antibody that becomes active only if a certain metabolite or a certain protein is above a certain concentration, and similar tricks that are based on very fine engineering on binding properties. We also take a very close look and pay a lot of attention to developability. So, for example, the antibodies that is now in the clinic, from the day we sent the sequence to the CRO until the day we had drug product after tox, after primary tox, it took 7 months. Typically, as you know, it may take 14 or 18 months, and the reason this was so fast is that the developability profile of this antibody was excellent, yields were very high, no aggregation, no PTM. It was very, very specific to the target, no tissue cross-reactivity, no ADAs in monkeys. So these are part of the parameters we're optimizing for. With that, let's look at the first example. The first example is an antibody against IL-2. And this is another important point about how we think about our discovery. Many biotech companies have the idea of looking for new biology. They say something like this: we tried to attack the old biology with a certain technology and it didn't work. So let's look for a new biology, a new target, and attack it with the old technology. This sometimes gives you great results, but in most cases, the new biology turns out to be not less complicated than the old one. What we're saying is, let's look at old biology. We understand very well why existing solutions are suboptimal and try to solve it with better technology. So instead of looking for new biology and attack it with old technology, we're looking for old biology and attacking it with new technology. And IL-2 is an old hat in that sense, that many people attack to cure cancer or treat cancer with IL-2. IL-2, as you may know, is a double-edged sword. It's a cytokine that can both activate the immune system and it can inhibit the immune system. It activates the immune system by binding to a trimer that is, by binding to a dimer that is expressed on cytotoxic cells. It inhibits the immune system by binding to a trimer that is expressed on Tregs. IL-2 loves Tregs much more than it likes T effector cells. What people suggested before is to engineer IL-2 such that it cannot bind the trimer and can only bind the dimer. This is a wonderful idea with one caveat. If you're successful, you give an engineered IL-2 and the engineered IL-2 is engineered such that it cannot go to the Tregs, then it goes to T effector cells. All the IL-2 in our body comes from cells that express the dimer. So if you're successful with this approach of engineered IL-2, then you lead inevitably and imminently to the secretion of more IL-2. And this IL-2 is not engineered, and it will prefer to go to Tregs. And this is actually what has been observed in the clinic. The first engineered IL-2 failed in phase 3 recently, and part of it is this negative feedback loop that is inevitable. What we were saying is, let's not engineer IL-2. Let's work with an antibody, and let's make the antibody grab IL-2 in a way that will prevent the binding to the trimer and will completely spare the binding to the dimer. If this works, we will lead to activation of the immune system. We will also lead to the secretion of more IL-2. But this will not be a problem. Why? Because there is a lot of antibody in the system. The antibody will grab the newly synthesized IL-2 and will send it again to the T effector cell. So instead of having this negative feedback loop that everyone else is having, we may be able to create a positive feedback loop. This is IL-2 in the middle, the red cytokine. You see it's a very small protein. It's bound to the trimer. What we want to be able to do is to be able to bind IL-2 in a way that completely blocks the interaction with CD25 and completely spares the interaction with the two other subunits, stabilizing the conformation that prefers the dimer and completely preventing the binding to the trimer. To do this, we employed our platform, and our platform always looks for an existing antibody as a template. In that sense, our platform is an in silico human immune system. The human immune system, when it needs to bind a new target, it always looks for a template that is an existing antibody. This existing antibody may be a germline sequence or a memory antibody. But the human immune system always starts with an existing antibody that may not have detectable affinity to the target, but is a good starting point. So our algorithm started with identifying an existing antibody that we predicted is a good template, but this antibody does not detectably bind human IL-2. Then we did all the magic I described before, and the algorithm suggested 3 mutations. You see the 3 mutations displayed here on the model. So we took a non-binder of human IL-2, we introduced 3 mutations. With these 3 mutations, the antibody now bound IL-2 with a single-digit picomolar affinity, and the binding was exactly where we wanted it to bind. We then employed the developability improvement algorithms and ended up with a sequence of human antibody that binds IL-2 exactly where we wanted, at single-digit picomolar affinity, and was developable in its profile. The antibody sits right there. You see the yellow protein is the antibody. Now look at some SPR data showing the specific binding of the antibody to the CD25 binding site. What you see here is an SPR where we first put the antibody on the chip, then we inject IL-2. You see that the antibody binds IL-2. Then when you inject CD25, right here, you don't see any binding, but when you inject the other subunits of the receptor, there is good binding. If you flip the order, still the same thing. So while the IL-2 is bound to the antibody, it cannot bind CD25, it can bind the other subunits. So this is data from human cells, and you can see that when you stimulate human cells and they start secreting IL-2, you treat them with the antibody. So if you just stimulate them, there is expansion of all the immune cells. But if you stimulate them and treat them with AU007, which is the name of our antibody, you see that you basically starve the Tregs on the left, but all the other cell types, the cytotoxic cell types, are expanded as if nothing is hindered, and in some cases, maybe we see even improved binding. Positive feedback loop we wanted. So what you see here is over time what happens, and you see that even 72 hours after the administration, after we stopped giving the antibody, we see that the expansion of the effector cells continues. The half-life of IL-2 without the antibody is measured in minutes. So what you actually see here are the first signs of this positive feedback loop we wanted to see. Looking at the inevitable mouse models, so you see here a model of the cancer, and it was treated either with IL-2 with IL-2 in red, our antibody in yellow, PD-L1, anti-PD-L1 in gray. So you see that the effect of our antibody is similar and maybe even a little better than the effect of anti-PD-L1. And when you combine the antibody and PD-L1 together, at day 17, when we stopped the study, most of the animals had complete clearance of the tumor, and the rest of them had already reversal, so that was very encouraging. And now we are treating patients with this antibody, and we hope to see similar results, but at least we have now some great news to mice. Let's talk about the next challenge, and the next challenge that we wanted to explore is the ability to have an antibody that can do more than one thing. Now, I'm not talking about a bispecific antibody. I'm talking about a symmetrical antibody with two identical heavy chains and two identical light chains, an antibody that has the sequence of a human antibody, that the somatic hypermutation on the VDJ sequences is sampled from the distribution of human somatic hypermutations in characterized repertoires. But we want to design it such that it can decide what it wants to bind at every point in time. Like we mentioned before, with the idea of being able to bind one cytokine under one set of circumstances and another cytokine under another set of circumstances. One case in which this could be helpful is Th1, Th2 inflammatory diseases. This is a process that involves many cytokines, relevant for diseases like atopic dermatitis, asthma, and many other diseases. Even some GI diseases are affected by this pathway. The pathway usually begins with upregulation of TSLP that leads to a sequence of cellular and molecular events and ends up in upregulation of IL-13. Indeed, both TSLP and IL-13 have been proven to be great targets. Antibodies against TSLP and antibodies against IL-13 have been approved in the clinic and became blockbusters quite instantly. However, about 50% of the patients, maybe even more, do not respond to these treatments. When you look at the pathway, you understand that if you block IL-13, you don't deal with other consequences of TSLP. And if you block TSLP, you don't deal with other causes that can upregulate IL-13. So what we want is an antibody that will know in every environment and in every point in time which cytokine is the dominant cytokine. So we design BD9, this antibody, and this antibody can bind either IL-13 or TSLP in both arms. It's a symmetrical antibody, but the paratope is engineered such that it can either bind TSLP like this or bind IL-13 like this. All right, so can it bind them well? So these are the affinities of BD9 to TSLP and IL-13 compared to the clinically approved anti-TSLP and anti-IL-13. And as you can see, the KD in both cases is slightly better than the reported KD of the clinically approved antibodies. So it's not just that we're binding both of them, we're binding both of them as tightly or even better than the clinically approved antibodies. We have data that show that we bind to the same epitope. I will not show it here because I'm running out of time. But do we have a similar function? So this is an assessment of the function of BD9 on cell-best assay compared to the approved anti-TSLP. And you see that the inhibition of the signaling with BD9 is slightly better than the inhibition of the signaling with the approved anti-TSLP. By the end of the talk, I'll figure out which button does what. Looking at human PBMC, so now we're taking human tissue, and on the human tissue, we want to study the effect of the three antibodies, the approved anti-TSLP, the approved anti-IL-13, and the, not yet in the clinic, BD9, okay? And what we want to see is the effect on well-accepted biomarkers. So CD23 is one marker for the effect of these treatments, and here you see the effect on CD23 expressing cells if you treat them either with the approved anti-TSLP, no effect on CD23, that's the red line, or the approved anti-IL-13, that's the blue line, or the BD9. Okay, so we have comparable effect to the approved anti-IL-13 on this biomarker. Another biomarker is TARC, so on the same cells, we are comparing the same three antibodies, and here you see that the approved anti-TSLP knocks down TARC all the way. The approved anti-IL-13 does not really affect TARC, and BD9 knocks down TARC as well as the approved anti-TSLP. So bottom line here is that we have one antibody that does both biochemically and in cell-based assays and in human PBMCs what each of the approved antibodies is doing, but in one antibody that can do it with both arms. The CMC and the development and the production of this antibody will be the CMC and production of traditional antibodies. It expresses well. It has very high specificity to these two targets. I don't have time to show this, but when we do all the standard polyspecificity assays, this antibody is specific just to two targets. Now I want to present some new ideas that we are working on using the same technology. So what I've tried to show before is that using the generation of our own data and then machine learning, we can come up with two important capabilities. A, precision binding, stabilize one conformation and avoid another, bind very surgically to a predefined epitope. And the second is to be able actually to design an OR gate. OR gate in mathematical logics or in computers is something that can either do this or that. And that's the TSLP-IL-13 antibody, and we have plenty of antibodies like this that we design. What we're working on now is what we call the exchangers. What are the exchangers? The exchangers are antibodies that can bind two different things. And in this example, they can either bind a small molecule or a protein. The small molecule could be a chemotherapy. And the protein could be a tumor-specific antigen. Why is this interesting? In the periphery, where the concentration of the tumor-specific antigen is low, and the concentration of the chemotherapy is high, such antibody will grab the chemotherapy and will do two things. Decrease the concentration of free chemotherapy in the periphery and extend the half-life of the chemotherapy from minutes to weeks, because now it's bound to an Fc-containing molecule. When this antibody swims around and gets to the tumor microenvironment, it sees its beloved tumor-specific antigen. So a classical competition begins, and a certain fraction of the antibodies will dump the chemotherapy onto the tumor microenvironment, doing two additional great things: increase the concentration of free chemotherapy in the tumor microenvironment, and attacking the tumor through the validated mechanism of the tumor-specific antigen. Another idea that we are working on, and we also have some nice encouraging results, is the idea of what we call super engagers. Super engagers are just like the engagers you know that can bring a tumor cell to a close proximity of an immune cell. But here we design an engager where each arm is a multibody. Each arm is an OR gate. So basically, the blue arm can either bind something like CD3, a marker on T cells, or CD16, a marker on NK cells. So the blue arm is an OR switch that can either bring one type of cytotoxic cells to the tumor or another type of cytotoxic cell. Note, it's an OR gate, so it will never create a situation where there is a synapse between an NK and a T cell that will kill each other. The orange arm is also a multibody that can bind one of two tumor-specific antigens. What's nice here is that you have one molecule, one CMC, one tox, but you actually create 4 engagements. You engage tumor antigen 1 to NKs, tumor antigen 2 to NKs, tumor antigen 1 to T cells, and tumor antigen 2 to T cells. So with this, you can actually really increase the therapeutic index of engagers and maybe cope with some of the toxicities that this approach has. With this, I obviously need to thank the amazing scientists that work with me on this. Many people said that their achievements depend on the giants on whose shoulders they're standing. So sometimes you also have the luck not to stand on the shoulders of giants, but also to work shoulder to shoulder with giants. And some of the scientists here are really brilliant thinkers and people that are a pleasure to work with. So I thank them and I thank you.